Hello, I'm Alden Thomas. I'm a pre-vet major, animal science at UMass Amherst, and I'm a sophomore, and I work in Laura Vandenberg's lab. And my research project is on the effects of TAML catalysts on white blood cell counts in male mice exposed as adults. For a little introduction to my research project, there are numerous sources of environmental contaminants in water, including both endogenous hormones and synthetic chemicals that mimic hormones. And elimination of these chemicals from wastewater is critical in maintaining a safe water supply and protecting public health. And one way to do this is through the use of TAMLs, which are tetramido macrocyclic ligands, and they are a peroxidase catalyst that is capable of degrading environmental contaminants, including low concentrations of estrogens. Our work focuses on new TAML, or NT7, and our work will evaluate whether NT7 has unanticipated hazards or not. In prior studies in the lab, we found that male mice exposed to NT7 for a 2-week period had decreased relative spleen weights. Based on these results, we hypothesized that this acute exposure would decrease circulating white blood cell count at the end of the exposure period. And the reason why we're looking at the immune system and to see if it's compromised or not by a TAML is because a decreased spleen size, the job of the spleen is one of them is to regulate the quantity of white blood cells in an organism. So if there's a decrease in the size or an alter of what its normal structure should be like, we could see a difference in the white blood cell count. So one way that we were able to look at all of this was we had, for our methods, we had adult male mice were co-housed with 2 females, each during a 2-week mating period. And during this time, they were provided with water delivering different exposures of 0.1% DMSO as our negative control, 1 mg per kg of NT7 as our low dose, 330 mg per kg of NT7 as our mid dose, and 1,000 mg per kg of NT7 as our high dose. We also ran 1 mg per kg of ethanol estradiol, or EE2, and 2,000 mg per kg of flutamide, or flute. And these blood smears were collected at necropsy, and in order to read the slides, they were first stained in a Wright-Gesman stain. This image in the center here shows what a typical stain will look like. As you can see, the white blood cells pick up this dye, and you can see all the white blood cells on the image are that darker blue. And on the right side there, you can see a little dark blue speck. Those are platelets. Those also will pick up some of the stain, leaving the red blood cells more translucent. They have a little blue coloration. However, they don't really absorb the stain. And this way we can easily differentiate between white blood cells and red blood cells. So that after they're stained, the slides are analyzed on an inverted microscope. And we also use a computer software called Zen computer software, where we're able to count and identify the red blood cells and the white blood cells individually. And what happens is per slide, there's 2 images taken per sample. And in one, it's each slide is a 40 time magnification. So when I get each image, there's a 1/20th of the image box placed on that image. And what I'll do is I'll count every single cell in that 1/20th of the image, and then I'll multiply it by 20 to get an average cell count for the entire field. Then I will write that number down, and then I will also write down the number of total white blood cells on the entire field. This is done twice per sample, which means that with our 25 samples, there was 50 total images taken. Now, when it comes to our results, we observed that there was no significant effect of NT7, EE2, or flute on either red blood cell count or white blood cell count in males following a 2-week acute exposure during the adulthood. As you can see here, is our average red blood cell count jitter plot and the average white blood cell jitter plot. And you can tell that unfortunately, the data isn't conclusive. It's a little bit all over the place. And in conclusion, while the data may have been Inconclusive, this set of animals is the smallest, with just 25 animals across the 6 treatment groups. This small sample size is because we only started collecting the blood smears from animals after we observed a small spleen size in the first set of animals. This study needs to be repeated with more adult males before we can draw a more definitive conclusion. So in the future, other analysis of the F1 generation of males exposed during gestation and perinatal period had decreased relative spleen weights at postnatal day 21. Our next step is to get red blood cell count and white blood cell counts from blood smears collected from these males. And I would just like to acknowledge that we are grateful to the other members of the Vandenberg Lab for their work on this project, and especially wanted to say thank you to Joshua Mogus and Jillian Zabo, who treated these animals and collected samples at necropsy. This work was also supported by an unrestricted gift from the SUDOC LLC. Thank you so much.